I want to turn it over to Scott Baraban from UC San Francisco. I've known Scott for many, many years. He is a fantastic researcher, has been one of the people who has pioneered zebrafish studies in epilepsy, and also been one of the rare examples who has taken that research and moved it not just from the basic science lab, but to the clinical as well. And he really listens to patient families, has done amazing work with the Dravet Foundation. I'm so happy to have him leading this session on the basic aspects of LGS and what's possible. Scott, thanks for being here.
Thanks, Tracy. And thanks everybody for getting up early here on the West Coast and the rest of you around the country. I want to kind of organize this session to be mostly interactive. As many of you already know, on the LGS model side, there aren't really, consensus ideas on how to build a good preclinical animal model of epilepsy, of LGS. And so I want to kind of, organize this around the three, I think, potential most cutting edge areas where people do model neurological diseases that patients actually have. And you're going to hear from three speakers who kind of address those different approaches and the really nice research they do. And what I want people to think about, and this is really for the discussion at the end, is how we can hone some of these talents and skills to actually come up with a reasonable or predictive, at least, preclinical animal model that will advance the field. And so I want to start with just a brief introduction. I'm not going to really present. I'm going to let the speakers talk about the science, but I want to kind of set the tone for how I think about this, at least. And so as many of you probably know, my laboratory uses a wide variety of animal models, everything from zebrafish, which you'll hear about, up through sea lions. And my view has always been that animal modeling or models for epilepsy is not a zero-sum game or winner-take-all. It's not that we have to have one specific epilepsy model and the others are no good. I think all the models have different contributions to how we think about science and how we think about studying these disorders. And the trick really is to fit the advantages of a specific modeling system to the types of things we're trying to model in patients. And so my approach has always been to start, like Tracy said, with learning more about what the patients are dealing with and thinking about the clinical kind of outcomes that we're trying to model. And so in LGS, of course, as you heard in the first day, this is a severe epileptic encephalopathy. And so you want to be able to model that somehow in your model systems. As we've talked about, and I think will be the theme today, it's a circuit disorder like most epilepsies, and there's very specific electrographic discharges that are specific to LGS patients, including the slow spike wave discharges you heard about yesterday and GPFAs. The other, of course, feature from a therapeutic perspective is that a valid preclinical model of LGS should also be pharmacoresistant. That is, it shouldn't respond well to available anti-epileptic drugs, and that would, in that case, that would mimic the problem that the patients already have. And I put this last one up because I think it's more for discussion of what other phenotypes the group thinks are potentially can be modeled in simple model systems, and where and how we go about prioritizing those and trying to figure out assays for those. Because I think, as was mentioned at the end of yesterday's meeting, the seizures and the epileptic discharges are one aspect of LGS, but there are certainly other aspects of comorbidities that we could also think about modeling in these systems, and really to prioritize those and brainstorm on how we would get there. So our approach here at UCSF in my lab has always been based on kind of an older theory that an animal model of epilepsy should mimic specific features of people with epilepsy. And the three features that we try to mimic are construct validity, which is basically how closely that animal model recapitulates the causal mechanism. So whether this is a brain injury or a genetic mechanism, this is what we want to start with, that our animal model has the same insult that the patients with that disorder have. The second is some form of face validity, namely how closely does that animal model recapitulate the actual phenotype. So if we're modeling a specific type of epilepsy where spontaneous seizures occur, and these are electrographic, we want our model to have electrographic seizures because that's what we're modeling, and that's the face validity the model should have. And the third part, which is we found in recent years is critical for moving things from preclinical models to the clinic, is the predictive validity of that model. So we define that as how closely an animal model recapitulates the treatment responsiveness. So if there's a standard of care set of drugs that those patients take, your animal model should also identify those drugs as being efficacious. And if there's a set of AEDs that patients don't respond to, your animal model should also not respond to those. And so those are the three kind of ways we look at modeling different types of epilepsies here in the lab. So the overall approach we've taken with zebrafish, which is the part of our lab that focuses on genetic epilepsies, is to basically generate a model that has the same cause. So knowing the different genes that cause these rare genetic epilepsies, a lot of these were discussed yesterday, and they emerged over the years from the EPGAP and the EPI4K types of projects. We've taken an approach using zebrafish to knock out those same genes using CRISPR technologies, and then look at the phenotypes in those zebrafish. And this is a culmination recently of about 6 or 7 years of work on this project, where we've now modeled or generated zebrafish models for 40 different known human epilepsy genes, loss of function genes, and those are shown here on the bottom. Each of these has been characterized in terms of their electrophysiology, their behaviors, their survival, a number of other features, and each of these are now available and published for any lab around the world that wants to use these to advance research on one of these gene disorders. So that if you go to the website listed here, you can find details on all the zebrafish lines that we've published. And if you're interested in working on one of them, just let me know and we'll send you fish. But I want to talk about, in this brief introduction, one specific example that we've made the most progress on, as Tracy alluded to, and that's Dravet syndrome. So for Dravet syndrome, about seven or 8 years ago, we started working with a Dravet model, a zebrafish model, that had a loss of function mutation in the same voltage-gated sodium channel that patients with epilepsy that Dravet syndrome have. This is a SCN1 channel, and it's very similar between zebrafish and humans. There's about 83% sequence similarity. It's expressed in the brain of the zebrafish, and it's a loss of function. So that fit our construct validity for generating a zebrafish model for Dravet syndrome. The second part was face validity. These zebrafish that have this mutation actually have spontaneous, unprovoked seizures. These can be monitored using simple behavioral measures where you're just videotaping the movement of the zebrafish larvae, as you see here on the left. They occur very early in development, so they have an early developmental onset, around 3 days post-fertilization. And you can quantify this by tracking this behavior in a well over time. And as you can see here from these two traces, a mutant fish that has this loss of function sodium channel has a lot of these seizure events within this well that can be tracked and measured. And the control doesn't have these events. More importantly, and most critical to how we ended up doing the screening for using this model, is if you record brain activity using a local field potential electrode, you can see that these animals have spontaneous electrographic discharges that we don't see in wild-type animals. These consist of what look like interictal or brief small amplitude events and larger ictal-like events that have much larger amplitude and are often followed by a post-ictal depression. These occur spontaneously. We don't do anything to the animals to elicit these types of seizures, and they originate in the central nervous system. We know this from doing calcium imaging studies. So that's the face validity. The other part of that is, beyond, like I said, beyond the seizure phenotype, these fish also have other phenotypes or other comorbidities that we see in patients with Dravet syndrome. They have an oculomotor deficit. They have a sleep disturbance, which you see here on the left in the sleep assay. They have a metabolic deficit that we work with Manisha Patel's lab to study. And they have early fatality or SUDEP. And so in this way, this zebrafish model mimics the two important things. It's a sodium channel mutation that people with Dravet syndrome have, and it has many of the same phenotypes that people with Dravet syndrome have. But what about predictability? Because if you want to do drug screening, you want to be confident that the drugs you identify have potential therapeutic implications once you found them. And so before we want to enter into a drug screening protocol, and the reason, of course, we use zebrafish is partly because we can do drug screening, because it's very easy to do hundreds of fish at a time. To start with, and before we got into that, we tested our fish using both the behavioral and the electrophysiological assays against known anti-epileptic drugs that are in the market that we already knew clinical data on. And so, as a pharmacoresistant form of epilepsy, most of these AEDs have no effect on spontaneous seizure behaviors or electrographic activity in our SCN1 mutant larvae. But interestingly, the drugs that are used as a standard of care for these patients, namely valproate, stiripentol, potassium bromide, benzodiazepines, and even in the form of the ketogenic diet, are all effective in suppressing seizure-like events in these zebrafish larvae. In contrast, if you look at a mouse model generated by Jennifer Kearney and focus on the spontaneous seizures, so the epilepsy in these animals, the mice are unable to predict these same types of AEDs that we use clinically. And so in this way, we think the zebrafish model has more predictive validity than the available mouse models. That led us, and this summarizes many years of work by a lot of very talented postdocs in the lab, to screen over 3,500 drugs using these assays in zebrafish. The drugs were all screened in a blinded fashion and were followed up were part of a two-stage phenotype-based screening. That means we look for drugs that stop the seizure behaviors, and those that stop the seizure behaviors also had to stop the electrographic activity. It's a two-stage assay because you can imagine a drug that stops a fish from swimming could also be a sedative or a muscle relaxant, so therefore a false positive. But those types of drugs wouldn't stop the electrical activity. From these 3,500 drugs we've screened over the 5 years, we identified 6 lead compounds. All of them had a similar mechanism of action, that they activated serotonin receptors, and all of them were effective in completely suppressing both the behavioral and the electrographic seizures in these zebrafish larvae. To show that the FDA actually finds this a valid approach of having this type of zebrafish model, all 3 of the compounds, which were repurposed drugs, were immediately approved by the FDA and given orphan drug designation for clinical development. 2 of those drugs, because they were already available in the market, we were able to test in small numbers of patients, including a study with Kelly Nutt at the University of Colorado, to show in small patient samples that are called compassionate use studies, that these drugs were efficacious in reducing seizure behaviors in people with Dravet syndrome. And so these drugs went straight from our laboratory discovery in zebrafish models to these patients. The bigger project is we're now developing the first compound called Clemozol. It's completed phase 1 studies. This is a very safe compound that used to be an antihistamine, and it's currently in a phase 2 clinical trial for Dravet syndrome, and it was recently approved for clinical trial studies in LGS. As a disclosure, of course, this work is done in collaboration with a company I've co-founded called Epigenics Therapeutics. And if you'd like to learn more about that, I'd be happy to write to me separately. And I'll turn you to those people and the ongoing clinical trials. But the big picture I want people to remember from this is that there's nothing that stops a laboratory from developing a novel model approach, showing that it's both valid and predictive, and then identifying new potential therapies that can go very quickly from your laboratory to patient trials and potentially help patients with that disorder. And so that's what I'm hoping we can figure out, a similar approach to generate LGS models that will allow us to do these types of studies. So in this this session of talks today, we have 3 excellent speakers. As Tracy said at the beginning, Allyson Muotri from the University of California, San Diego, who will talk to us about human-derived organoid approaches to model human diseases, neurological diseases in particular. This is a very exciting technique that many of you have probably seen, and you're going to learn a little bit more about it this morning. Megan Dennis, a very talented young professor at the University of California, Davis, who's developing zebrafish models for a number of genetic disorders and showing various ways you can measure comorbidities in zebrafish. And you'll hear a little bit about her work and some of the new genes she's discovered and modeled. And then Min-Shan Xu from Baylor College of Medicine, who hopefully will avoid the incoming tropical storm in Texas and get to his office in time to tell us about a rodent or a mouse model, in this case, of a rare genetic epilepsy and electrophysiology and behavioral studies he's done in that type of animal model to mimic SCXBP1 disorder. And then at the end, we'll come back around to hopefully have a larger panel discussion where we can integrate some of these ideas and get some kind of, maybe not a consensus, but at least some ideas on where we can move the field forward after this.